This is Lesson 2-10, Principles of Automated Hematology. We've been counting blood cells since around 1855. At that time we were using a counting chamber called a hemocytometer, and we used this until around 1950 or so. This is a picture of a hemocytometer, a microscopic view. And the way this is used, we dilute cells in a saline or acetic acid and use a pipette to pipette the sample onto a grid. And after the cells have been allowed to settle for a period of time, we are going to count areas on the grid. We're going to count cells in certain areas on the grid. And we still use the hemocytometer today, but only for backup of automated hematology or automated cell counting. A little bit about the history of cell counting. First we'll start with the Coulter principle, and it was around the 1950s, 1956, that William Coulter invented the first automated method of counting cells. He was an innovator, an entrepreneur, and an inventor, and he invented what's called the Coulter principle. And the Coulter principle is a reference method for counting and sizing particles or cells that are suspended in a fluid. Once this was developed, it led to major breakthroughs in science, medicine, and industry. Later on, Coulter partnered with his brother to form the Coulter Corporation, which led to a global diagnostic company that was created for more than 40 years. And around the time of his death in 1997, the company was sold to Beckman Instruments and then became known as Beckman Coulter. And you'll probably see that term on some of your analyzers in the laboratory, or that name, Beckman Coulter. Just as a side note, Beckman was also a prolific inventor and a chemist, and he was actually a native of Illinois, specifically Columental, Illinois, and he invented the pH meter and the spectrophotometer, which we still use today. The spectrophotometer is actually the basis for most of our chemical analyzers. So we used the Coulter counter principle until the 1970s. However, this type of technology was only counting our white cells and our red cells. Our platelets we determine manually by counting on a hemocytometer or a blood smear. And then hemoglobin values were determined manually. We can get a hematocrit level by using the hemoglobin and a red cell count. So we did this until other types of cell counters were developed. Automated cell counts are actually more reliable than manual methods. Chance of error, or the coefficient of variation for automated cell counts, is 1%, and manual counts are coefficient of error or variation is around 10%. There are two basic types of cell counters that use electrical impedance and light scatter cell counting. So we'll start with electrical impedance first. And in electrical impedance, cells are diluted in an electrolyte solution. And just keep in mind that an electrolyte solution is able to conduct electricity. So as cells move through the entrance of the counting chamber, and that's called the aperture, cells will break the current of electricity or impede the electrical current and are counted according to size. White cells are larger than red cells, so the impedance will be very large and are larger than a red cell. Platelets are the smallest of all our cells, so that electrical impedance or breaking of the current will be smaller, and that's how those are counted. Counting chamber here. You'll notice the electrodes in the diagram. The external electrode has a positive charge and the internal electrode has a negative charge. The electrolyte solution is going to flow from positive to negative electrode, and that's what pulls the cells in with it through the aperture tube. And you see the little place where the aperture is, how it pulls the cells in towards the negative charge. And since blood cells are very poor conductors of electricity, as each cell moves past that aperture, an impedance or an interruption in the flow is created and the cells counted. Your electrical impedance analyzers produce a three-part differential. Three-part differential is lymphocytes, monocytes, and granulocytes. And an example of a histogram showing three-part differential: your lymphs, your monos, and your granulocytes. Made a lot of improvements in our hematology technology over the years. Counting of cells using electrical impedance did have some inherent problems. For example, if cells passing through the aperture did not pass through the center of the aperture, they were actually measured larger than their size. Or if cells got trapped in that little aperture hole, they were counted over and over and over, falsely increasing the count. So the cell counter was modified so that the flow was channeled to the center of the aperture, and then the information they obtained was more accurate. And analyzers were able to create actually a graph, as I showed you a histogram before, to show the actual cell size on the X and the Y axis. Here we have an example of a light scatter type of technology. Light scatter cell counters use a laser beam or a halogen light beam, and it's directed at the stream of blood moving through the aperture, and cells actually are narrowed into a single file. The light beam strikes the cell and it's scattered at an angle. And each type of blood cell has its own angle depending on its size and the cell that's counted. Light scatter cell counters produce a five-part differential, and I have an example of a five-part differential report later on in this audio lecture. There actually are two types of light scatter technology. There's a side light scatter and a forward scatter. Side light scattered is where light is scattered at a 90-degree angle from the cell, and it defines the internal complexity and granularity of the cell. Neutrophils and eosinophils produce a great deal of side scatter because they have granules. The forward scatter technology has a light that continues in a forward direction and relates actually the cell size. Large cells such as monocytes and neutrophils, they produce more forward scatter than a red blood cell or a normal lymphocyte. Here's an example of a light scatter. See how the laser hits a single particle and then it scatters in different directions. Once the instruments gather data, it's going to send it to the computer software, and it's going to graphically represent the cell population. You notice the x-axis on this graph represents the cell's internal complexity or granularity that I'd mentioned before. X-axis is labeled complexity, and that's determined by the amount of side scatter. The y-axis that you see here, that's labeled size, separates the cell populations and is based on the cell size, and that's determined by forward scatter. Three other types of automation actually that's more in use now. You have flow cytometry, and then we have cytochemical or fluorescent staining, and then image processing. So we're going to briefly cover those next. Here you have an example of a flow cytometer, hematology analyzer, and the purpose of flow cytometry is to detect and measure multiple properties of cells It identifies and then quantifies or counts the cells. Here we have the internal workings of the flow cytometry. Not a lot of difference, really, when you look at it, except for the type of light source that's used. Um, When I say there's not a lot of difference is because our cells are put into a suspension of fluid, and they're channeled through the inner column of the flow cell. The outer column of the fluid controls the diameter of the sample column so that it can narrow and isolate single types of particles or cells. Single particles or cells are going to pass through a laser beam. The light scatter is detected by the photo detector. You can see where that's located. And these instruments are going to use light scatter to create a 5-part WBC differential. Here you have an example. Though it's kind of small, but this is what a 5-part differential looks like on a computer screen of an analyzer. Thanks, Samsung. Here's an example of fluorescent testing. And we can attach these fluorescence compounds to the sample, and they help us to differentiate cells. Actually, this type of analyzer can do immunophenotyping. It can do white blood cell differentiation and counting. It can do reticulocyte counting and also DNA analysis. When we're using fluorochromes, uh, this type of analyzer uses a laser beam and has a specific wavelength of light, and it's going to excite the fluorescence or the fluorochrome that is attached to the cell. The fluorochrome is going to emit a light at a longer wavelength. The light is then detected, measured, and quantified. The intensity of the emitted light is proportional to the amount of fluorochrome attached to the cell. And your computer software analyzes the data and then generates a scatter plot. Okay, last type of technology we're going to discuss is automated differential counting, and this processes images in the analyzer. This type of analyzer is going to store thousands of images from normal peripheral blood cells. And if you remember, peripheral blood is venous blood. The instrument's going to scan the blood smear and compare the cells with the images in its memory. This machine does have some downsides to it because it does rely on a well-made smear, has to have proper cell distribution on the smear, and proper staining is essential to this type of analyzer. As always, when using analyzers, we're going to follow safety rules regarding standard precautions, and use of your personal protective equipment is essential. There's oftentimes you're going to have to take the caps or tops off of your tubes of blood, so wearing gloves and covering your clothes is important. And each lab is going to have its own standard operational procedures for their own individual hematology analyzers. If you remember, your manufacturers do provide calibrators controls and also specify the frequency of when you should calibrate and use controls. A lot of our labs use an inexpensive method of quality control, and this involves choosing maybe 5 or 10 samples from a day's workload and that have results that are within normal reference ranges, and that they reanalyze these throughout the day. Others use manufacturing controls to run throughout the day, um, and also reanalyze patient samples to watch for trends or shifts with the analyzer. Hematology analyzers are a little bit more different than your chemistry analyzers. Chemistry analyzers, you can run controls essentially usually once per shift. But with hematology analyzers, they need to be checked periodically throughout the day, and you should follow standard operating procedures for the type of instrument that you're using.